We will now start our discussion on the flowering process of plants, which is covered in Module 1 of our syllabus. For this lecture, we will be talking about the developmental phase transition in plants and the signals that regulate this transition, particularly from the vegetative to the reproductive phase.
Like animals, plants also undergo a series of developmental stages. A mature cotton plant, for example, will start out from a seed. A newly germinated seed will be composed of the embryonic leaf, known as the cotyledon, the embryonic root, known as the radicle, and the embryonic stem, known as the hypocotyl. With time, the seedling will develop new leaves and stems. This will be accompanied by lateral growth in the form of branching. After several weeks of vegetative growth, the plant will start producing flowers. These flowers will then be pollinated to produce the balls, which will then produce the fiber. Within a period of 130 to 160 days, the cotton plant will complete its life cycle. In general, these different stages in the growth of a cotton plant can be divided into two key developmental stages or phases, the vegetative and the reproductive stage. The reproductive stage is characterized by a rapid increase in photosynthetic capacity that results in rapid increase in the size and mass of the plant. The vegetative stage can further be divided into the juvenile vegetative phase and the adult vegetative phase. When the plant transitions from the juvenile to the adult vegetative stage, there are abrupt changes that we can see in the physiology as well as in the morphology of the plant. These observable changes are referred to as heteroblasty. This slide shows you examples of heteroblasty in two different plant species. The first example demonstrates changes in the leaf shape of Passiflora edulis or passion fruit as it transitions from the juvenile vegetative to the adult vegetative stage. In particular, this panel shows you leaves that were collected throughout the life cycle of the plant. Notice in this leaf a slight protrusion as indicated by the red arrow. This is the beginning of what is eventually to become a lobe in the leaf. This morphological change marks the transition of the plant from the juvenile vegetative to the adult vegetative stage. From this point on, until the leaves become fully trilobe, the plant is at the adult vegetative stage. The stage before that, when the plants only have monolobe leaves, is the juvenile vegetative stage. Another good example of heteroblasty is the changes that can be seen in the leaves of Arabidopsis thaliana as the plant transitions from the juvenile to the adult vegetative stage. At the juvenile stage, the leaves of Arabidopsis have longer petioles. The petioles are the stem-like structures that connects the leaf to the plant. The leaves are also rounder, with smooth leaf margins and trichomes or hair-like structures on the lower side of the leaf. As the plant transitions to adult vegetative stage, the petioles become shorter and the leaves become larger and also elongated. There will also be serrations or teeth-like structures in the margin of the leaves, as well as trichomes in both the upper and the lower side of the leaves. Aside from morphological differences, there is a key physiological feature that differentiates a plant at the juvenile and adult vegetative stage. At some point in the life cycle of plants, they become reproductive or develop the ability to reproduce. This switch is induced by changes in the environment or by chemical signals within the plant. We will be discussing this in more detail in succeeding lectures. Plants at the juvenile vegetative stage are insensitive to these environmental as well as endogenous cues. In contrast, plants at the adult vegetative phase are receptive of these signals. What does this mean? When a plant at the adult vegetative stage perceives signals from the environment, such as changes in temperature or day length as the season turns from winter to spring, it is able to process those changes as cues to switch from the adult vegetative to the reproductive growth. The reproductive stage is a key phase in the plant life cycle when it develops the ability to reproduce. During this phase, the plant starts to bear flowers, which are the reproductive organs of the plants. These flowers will eventually produce the seeds through the controlled process of pollination and fertilization. The amount of time required for plants to make the transition from the adult vegetative to the reproductive phase is species specific and depends on the life cycle of plants. Arabidopsis, for example, has a short life cycle of 6 to 8 weeks. From planting, Arabidopsis will grow vegetatively for 4 to 5 weeks before it transitions into the reproductive phase. Rice takes approximately 80 to 120 days to complete its life cycle. The transition from the adult vegetative to reproductive stage occurs in about 60 days. Carrots are biennial and will not flower in its first season. It will just grow vegetatively. But if there is a sudden change in temperature and carrots are exposed to unusually warm weather, they can be tricked to put their energy into flowering. Trees which have life cycles extending up to more than 100 years like citrus and poplars make this transition between 5 to 10 years. This is the reason why orchards will usually plant grafts or stem cuttings to have the trees flower and become productive in less than the time the plant species required to become productive if it was grown from a seed. The transition from the adult vegetative to reproductive stage is accompanied by a change in the shoot apical meristem into inflorescence meristems, which are then converted to floral meristem. To better understand these changes, let's define a few important terms. From your basic biology, recall that the meristems are undifferentiated tissue that are composed of actively dividing cells known as the meristematic cells. The meristematic cells are undifferentiated cells in plant meristems that are undergoing rapid division. They are just like stem cells and can develop into various organs or plant parts. Meristems that are located in the apices of the plant, like the roots or shoots, are called the apical meristem. The apical meristems are responsible for primary growth. The shoot apical meristems give rise to organs like leaves and flowers, while the root apical meristem give rise to roots. Aside from the apical meristem, plants also have lateral meristems. The lateral meristems are composed of the cork cambium and the vascular cambium and allows the plant to grow laterally. That is why they are called lateral meristems. An example of lateral growth is the increase in diameter. Now let's go back to what happens to the shoot apical meristems during the transition from the adult vegetative to the reproductive stage. Throughout the duration of the vegetative stage, including the juvenile and the adult vegetative stage, the shoot apical meristem remains as it is and give rise only to leaves. As the plant transitions from the vegetative to the reproductive phase, the shoot apical meristem also transitions from a shoot apical meristem to an inflorescence meristem. In doing so, the plant gains the ability to produce not only leaves but also flowers, which are the key organs for sexual reproduction. Now, the inflorescence meristem are composed of individual branch meristem, which develops into two spikelet meristem and then two floral meristem. We will discuss these processes in more detail in our succeeding lectures. Right now, I just want to give you a general idea as to the necessary changes that need to happen within the plant so that it can complete its transition from the vegetative to the reproductive stage. To summarize, plant development towards seed production progresses through two distinct stages, the vegetative and the reproductive stage. The vegetative stage is characterized by a rapid increase in size and mass of the plant. The vegetative stage is further subdivided into the juvenile and adult vegetative phase. When the plant transitions from the juvenile to adult vegetative stage, abrupt changes in the morphological and physiological properties of the plant occurs. These changes are referred to as heteroblasty. The shift from the adult vegetative to reproductive phase is marked by a switch in the identity of shoot apical meristem to inflorescence meristem, which in turn develops into floral meristem.